Hello, in this video we will discuss about the colorectal cancer pathogenesis, the whole mechanism. In this video we will learn. So what is the colorectal cancer? Basically it is the colon cancer which that occur in the large intestine, remember. So the large intestine is also known as the colon. So here is the esophagus, and the digestive system is the second part of the body digestive system, and this is the small intestine will open into the large intestine, and here is the stomach. This is the esophagus used for the swallowing of food into the stomach, while this is the large intestine and small intestine, and this is the appendix and the anus, rectum to anus is used for the defecation, while the ingestion through a oral cavity to the esophagus and the stomach will begin the digestion. So here is I am drawing the bigger picture of the large intestine. Here is the transverse colon, and this is the descending colon, and this is the ascending colon, and this is the appendix, and this is the rectum and anus. The basically here is the sigmoid colon also present. So this is the rectum is store waste, and anus is used for the defecation. So the cancer can cause with the highlighter in the yellow color. So the stage 1 is nothing, the rapid any change but mutation cell division in DNA lead to preparation oncogene activation due to the DNA methylation, a DNA damage or the break of the DNA or other problem during the mismatch repair will incomplete due to the absence of enzyme. Or on the other hand, there is another process which that is the point mutation are occurring in the stage 1. So anyhow, the stage 1, before the stage 1, the stage 0 phase also occur. So the stage 2 is the carcinoma formation. In this way, remember the carcinoma mean the tumor formation and tumorigenesis and angiogenesis. Genesis due to the growth factor will be bound with the cell, will rapidly divide due to the uncontrolled cell division due to the oncogene activation like ras and mice gene. So in this way the stage 2 can be cured and stage 1 also can be cured, but the stage 3 is also the carcinoma progression. In this way the stage 3 is also can be removed if the detection earlier. So the tumor formation and tumorigenesis and the angiogenesis and the growth factor is involving. After this basically is the uncontrolled cell division will decrease the size of the cell while the increase of the number of the cell, and stage 4 which that is the metastasis. In this way the stage 4 is the malignant tumor formation and split into cell and moves into blood due to the smaller in size after the angiogenesis. So in this way the permeability of the cell will easy due to the inflammation and the easy to move, and the result in the different tissue and organs for the further cell division to progress the metastatic path. A metastatic stage will lead to uncure. So this is the large intestines contain epithelial cell, and this is the normal epithelium. Remember when we will overeating chronically and bad eating habit or the genetic or family history or genetic mutation or radiation which that will lead to cause cancer in the epithelial cell. Epithelial cell means the damage of the DNA. So due to the damage of the DNA, which that will lead to uncontrolled cell division due to the oncogene activation, due to the methylation. So in this way, here is you can see this is the cell which that is damage of the DNA in the nucleus will lead to transformation tumor initiation. In this way, it is the stage 0, which that will lead to vent catenin, beta catenin, which that went beta catenin pathway. In this way the one of that which that is oxygen attached with the APC and the ZNRF3 which that is a subunit with the receptor, will bind with the Vent as a ligand, will lead to inactive the glycogen synthase kinase 3 beta, inactive to activate. And on the other hand, the hyperactivation will lead to Vent beta catenin formation. This is the beta catenin is the transcription factor, will bind with the DNA, will lead to activate the mice and CCN and P1 gene activation will produce, as well as cyclin and cyclin dependent kinase will increase, will lead to uncontrolled protein enzyme production, will lead to release the pro-inflammatory cytokines due to the inflammation, as well as inflammation, also the inflammatory cytokines through beta catenin due to the response of the DNA damage, and the uncontrolled cell division due to the growth factor increase in the cell abnormally. So here is the pathogenic bacteria: E. coli, F. nucleatum, and B. P. fragiles, which that will be target to the cell, and in this way the genotoxic metabolites. This genotoxic metabolites, the loss of surface bacteria, the loss of surface bacteria barrier dysfunction, which that will lead to permeability of this bacteria. Bacteria is enter into the mucosa, from the lumen into the mucosal pass through a mucosa. Inside of the epithelium, not mucosa, while the mucosal membrane is the upper. So invasion of the commensals bacteria and pathogenic species will enter. In this way, the myeloid cell, which that will be triggered by this, will produce the interleukin 6, 12, A, and 22 and 23. After this, the progression of the loss of surface barrier function will lead to epithelial tumor formation. This tumor formation, which that is coating with a bacterial biofilm, and in this way, the different size of the cell will divide unequally, will change the cell structure and the cell function, not like a epithelial cell, because the DNA will damage. And in this way here you can see the metastasis stage 4 will be occur. In this way the cells will be moved into the blood circulatory system, and where they can easily reside in the different tissue to progress the stage 4 of cancer. So in this way here is the commensals and pathogenic species of the bacteria will also inhibit or trigger the immune cells, like a natural killer cell inhibition. So in this way the natural killer cell when inhibit, in this way the cancerous cell will not kill by the natural killer cell, and the T regulatory cell trigger will mean the regulation of the cancer cell progression and cell division more. In this way, the uncontrolled immune system will occur due to the inhibition of natural killer cell. And MDSC and CD8 T cell, which that is also inhibit. In this way, the inhibition of CD8 positive T cell, the cytotoxic T cell will lead to inhibit the killing of the cancerous cell. So in this way the cancerous cell will independently replicate, and the T cell is important for the T regulatory cell will regulate and control the cell division of the oncogene containing uncontrolled cell division will be progress, and the immature dendritic cell will also, and the Tn and Tm will also trigger go into the blood circulatory system, will lead to metastasis and inflammation. And in this way, remember that thing, the CD8 and natural killer cell will inhibit, and the T helper cell or T regulatory cell will be stimulate, will be regulation to promote the metastasis, promote the stage 4. That is why the natural killer cell and CD8 cell will not functional. In this way, the inhibition through commensals and pathogenic. In this way, you can understand the interleukin 6 and 12A and 22 and 23, which that will promote. So the mycotoxin also produced from the fungi as per genus, species, Penicillium and Fusarium, which that is also mycotoxin, which that will lead to tumor progression. So remember also permeability into the across the border of the epithelium will move, and the normal epithelium is here, and this is the abnormality due to the inflammation will trigger to the antigen presenting cell. So this is the intestinal metaplasia formation will occur, and in this way you can understand this is the antigen presenting cell which that produce the interleukin 17A and 22, which that due to the inflammation and the invasion of the mycotoxins, this mycotoxin will stimulate the antigen presenting cell. This antigen presenting cell which that will stimulate the T regulatory cell and other things. Basically, it is normally it can be killed through antigen presenting cell will stimulate the natural killer cell, and the CD8 T cell will lead to kill the progressive cell. But in this manner, the natural killer cell and the CD8 T cell will inhibit via a commensals and pathogenic species, which that will lead to progress at stage 4. So this is the colorectal cancer pathogenesis. I hope you make sense about this. Basically, this is the briefly overview about the colorectal cancer with the mechanism. So I hope you make sense. Please make sure to subscribe, like, and share if you like this video. And thanks for watching. Bye.